And hello guys, and today we're going to do chapter 5 of Campbell's biology, and I'm going to honestly keep the introduction brief because chapter 5 is a lot, and sorry for not posting this sooner or later. These are essentially my unit 1 notes, and now we're on chapter 5, which is the last unit of unit 1. Well, chapter 1 is not really a part of unit 1, but whatever. But yeah, sorry for not posting this earlier. School started, so I had some things to do, and yeah. So let's just start off with chapter 5. So chapter 5 is a long chapter about the structure and function of large biological molecules. Now, essentially, the first term you need to know, are like polymers. So essentially, polymers are essentially long lengths of repeating monomers. And so monomers are just the repeating building blocks of polymers. So polymers are pretty broad. But you do need to note that lipids are actually not considered polymers or macromolecules. We'll get into lipids in just a minute, but apparently they're not like big enough to be considered polymers and macromolecules. Just note that they're not considered them. It doesn't really matter why, but just know they're not. And so the three polymers you need to know are obviously carbohydrates, proteins, and nucleic acids, and we'll be talking about their intricate properties in this video. And like I said, if you go down, monomers are simply just the repeating building blocks of polymers. Now, the two most important reactions in all of biology, and it's particularly biochemistry, if you want to be specific, it's not even just in biochemistry, but the two most important reactions you need to know, not only for chapter 5, but for the rest of this book. They're just first introduced in chapter 5, but the two main reactions you need to know are dehydration reactions and hydrolysis reactions. Now, dehydration, and I think these names are pretty suggestive, so they won't be hard to forget. Like, for example, dehydration reactions are essentially reactions that form two monomers together due to the loss of a water molecule, not to the addition. So basically, dehydration to like losing water molecule, and this essentially forms bonds. And you can see in the picture, right? The dehydration reaction, essentially you have this unlinked monomer that loses a water molecule, and then the monomers form together. Now, hydrolysis is basically hydro means water, lysis means breaks. So essentially, the addition of a water molecule that essentially breaks the linkage between two monomers. And so these reactions are very important. They're honestly included in almost every reaction, especially in biochemistry and especially in metabolism. You'll see these reactions in almost every single thing in metabolism, like literally. So they're very important to understand. So now carbohydrates are basically, just need to know, they're the sugars of life, right? Now, basically a monosaccharide is basically the monomer unit of a carbohydrate or sugar. So a disaccharide is essentially two monosaccharides connected by a glycosidic linkage. And this is obviously due to dehydration reactions, because obviously if you have these monosaccharides joining together, you have them joining together because of the removal of a water molecule. Now, glycosidic linkages are essentially just linkages between two monomers that connect certain carbons of monomers together. So, for example, they might put you might see reactions say like 1,4. They just mean that the first carbon is connected to the fourth carbon. So just glycosidic linkages between two monomers that connects carbons of those monomers together. And these obviously connect monosaccharides, right? By dehydration reactions, as we learned in the previous term. Now, glucose is essentially a monosaccharide. That essentially is a hexose, so a six-containing carbon sugar, and it's an aldose because its carbonyl group is on the outside of a chemical skeleton. Now, for chapter 4, if you didn't see it, check my chapter 4 video. We talked about functional groups, so I'm gonna kind of assume you understand functional groups. And so basically, the chemical formula is C6H12O6. Now, galactose is like literally like slightly different, like really slightly different, but that slight change in structure changes a lot. So galactose is basically a monosaccharide that is basically the same thing as glucose. It's just a structural isomer of glucose, and it's also an aldose because the carbonyl group is on the outside of the chemical skeleton. Now, fructose is a ketone, so that means it's basically the same thing as glucose and galactose, but it's a ketone, which means the carbonyl group is within the chemical skeleton. And so you can see in the picture over here, you have glucose, you have the carbonyl group on the outside because they're both aldehydes, and then you have fructose, the carbonyl and galactose, the carbonyl groups on the outside, and then you have fructose, which the carbonyl group is within the chemical skeleton, making it a ketose, and it's sometimes referred to fruit sugar. And also, you can just see the differences. You can see in glucose, the hydroxyl and hydrogens are switched in glucose and galactose. So just know they're structural isomers. I honestly recommend that. There's obviously a lot more sugars, but these three are the most important for you to know. In fact, I'm pretty sure they're considered the three dietary sugars, but that wasn't added in Campbell, so don't worry about that. But yeah, I would recommend like knowing how to like draw these. So definitely focus on remembering these and how to like draw the chemical skeletons. They're quite simple because it's just glucose, and it's also important because it's used in every reaction, right? It's using a lot of metabolism, so it's definitely important to know those. Now, lactose is basically a glucose plus a galactose. I think this is pretty easy to memorize, and then you just need to know that they're in 1 to 4 glycosidic linkages. So basically, 1 carbon to the 4th carbon, and that's the chemical structure C12H22 and HL11. Now, sucrose is just fructose plus glucose, but in a 1 to 2 glycosidic linkage. And so these are just disaccharides because they're just obviously two monosaccharides that are joined by glycosidic linkages. And I would say that don't focus on memorizing like this part over here. If you want to, go ahead. It's really important. But I feel like once you get to like a biochemistry textbook, then probably memorize them. But for now, just get familiar with them. Don't spend too much time memorizing it because you're probably going to forget it. So the best way to memorize something is by applying it. So you're going to be applying these chemical skeletons a lot later in biochemistry textbooks and all that. And once we get into like those textbooks. Polysaccharides are essentially these large macromolecules consisted of like thousands to few hundreds of thousands of monosaccharides connected together by glycosidic linkages. Now, I'm about to talk about four main, I think like 3 or 4 main storage polysaccharides, but I'm only going to briefly talk about them because there's later chapters in this book, like the plant chapter about like the structure of cells. Will obviously talk about cellulose, so I'm not going to talk about it specifically here. I'm just going to talk about it in brief terms because yeah, this is chapter 5. It's not about cellulose. So just know that for chapter 5, all you need to know is that cellulose is basically polysaccharide that strengthens the cell walls of plants. Then glycogen is basically energy of glucose in animals. And then starch is a polysaccharide that stores glucose for plants. Now there's two storage polysaccharides for starch. The first one is amylose, and amylose is basically the unbranched version of starch. And unbranched just means that there's no like branches. There's like it's basically just like a continuous like linear chain. But then amylopectin is basically the more complicated version, and it's like the branch version. So basically, there's like branches. It's not like a linear chain. It's just a branch, and basically at the branch points you have 1 to 6 glycosidic linkages. So amylopectin, it's branched. Amylose is unbranched, and 1 to 6 glycosidic linkages. Like I said, memorize that. It's not really a big deal. Just memorize it. Sometimes it just stays in your head. And then a chitin is basically the polysaccharide for the exoskeletons of arthropods. Arthropods are like insects, so we'll get to that stuff in the animal unit, but it doesn't really matter. Okay, so fatty acids are basically don't really worry about those storage polysaccharides for now. Honestly, throughout chemical biology, you'll see their usefulness in more intricate terms than this. Now, fatty acid is basically just a long carbon chain and basically like 16 to 18 carbons. And so at the end of the skeleton, it has a carboxyl group. Remember, carboxyl group is like COOH, and essentially it acts like an acid, so it picks up hydroxyl groups, which essentially reduces the hydroxyl concentration and increases the hydrogen ion concentration, which makes it an acid. And so basically, fatty acid is basically this long chain of carbons, and then the carboxyl group is at the end, giving it the name fatty acid, right? So I think that's pretty simple to remember. And then glycerol is basically an alcohol with 3 carbons that bear a hydroxyl group. And if you want to search these chemical skeletons, I actually recommend you do. But in these overviews, we just talk about the terms. And so I recommend actually before you watch my overviews, you read the chapter and then you look at my overviews, because when you look at the overviews, you might have like stronger, like more chances to memorize it. But yeah, actually watch the overview before you read the chapter. Watch my overview before you read the chapter, but that doesn't matter because watching so you have an idea when you read the chapter. My bad. So saturated fats are basically fats that only form single bonds in their hydrocarbon chain. Generally, because they only form single bonds, so they don't, so that their hydrocarbon chain is pretty full. They're tightly packed. so they're solid at room temperature, making them bad for you. So an example of this is like butter. Butter is a saturated fat, and it's like basically has no, it only forms single bonds in its hydrocarbon chain. That's probably the most important thing to know. Single bonds, and it's bad for you. And then unsaturated fats are like fats that form one or more double bonds in their hydrocarbon chain. Now, cis unsaturated fats. I think we talked about this in our was it chapter 4? I think we talked about this though. Cis bonds are essentially bonds that share the same valencies on the same side about a double bond. So basically, cis bonds uh, make kinks or bends to unsaturated fats, which prevent the fat from packing tightly or becoming a solid at room temperature. Therefore, they're generally liquid at room temperature, and this makes them good for you. So yeah, that's all you really need to know. Trans fat is, as you probably know, is really bad for you, and this is because their valencies are on opposite sides of about the double bond, and these fats usually are solid, so just like saturated fats, and pack at room temperature, and they make them bad for you. Um, okay, and yes. So steroids are essentially like lipids that have four similar fused rings. An example of this is cholesterol, and so yeah, that's pretty simple. And like we're just like briefly touching these concepts because like there's a lot of like broad stuff, and like you'll see later in the chapters there's a much more specific things. But I think the first unit is just like an introduction, just broad terms. Phospholipid is basically two fatty acids plus a glycerol, and so there's a hydrophilic head. I probably should have written that down, but I guess I will. There's a hydrophilic head and a hydro and two hydrophobic tails. Two hydrophobic. I can't. Okay. So yeah, basically all you need to know. And then also like I should have probably added this, but I was too lazy because I actually wrote all of this by myself, typed it up, no copy and paste. That's the only way how you memorize. Basically the phospholipids help the cell membrane. They're the main constituents of the cell membrane. That's actually a very important thing to know. And they actually form bilayers, but do not worry about this. As we go to our cell unit, you'll learn all about phospholipids and stuff. Like I said, don't really worry about this too much. Just know that phospholipids means two fatty acids plus glycerol, and they're main constituents of uh, cell membranes. And they did talk about the bilayer in the chapter, but like I said, you're already gonna see it in the cell unit, so it's not really too important to know that because you're already gonna read it about it again in the future. So don't focus too much about it. Just focus on the one idea of two fatty acids plus a glycerol. Now, an amino acid essentially the building block or monomer of proteins. Now, amino acids consist of one asymmetric carbon called the alpha carbon with one free hydrogen, and then one R group, and then one amino group and a carboxyl group. There are 20 amino acids in biology. Amino acids differ by their side chains. Some are more complicated than others. Their side chains can also be hydrophobic and hydrophilic. I actually probably let me just draw it out for you. So let me go on whiteboard. Wait, no, Microsoft Whiteboard. Let me just draw it out for you on Microsoft Whiteboard. Sorry. So yeah, I'll just draw it out for you so you can see what I am talking about. Yeah, because I want you to know what a amino acid looks like. Actually, no, you know what? I won't draw it. Oh yeah, it's right here. My bad. So this is what an amino acid looks like. As you can see, the asymmetric carbon is here, and the R group is there, the hydrogen's there, and the carboxyl group is there. Basically, honestly, if you know functional groups from chapter 4, if you memorize them, then knowing the structure of amino acids should be pretty simple, fairly simple for you. But yeah, that's all the information you need to know. And then a peptide bond is basically the covalent linkage of carboxyl groups. So basically, there's okay. So basically, we all know that like amino acids have carboxyl groups. That's just stated. So essentially, when these carboxyl groups become adjacent to each other, they essentially form a peptide bond. And this is obviously due to dehydration reactions, right? As you can probably assume. And then okay, this is supposed to be polypeptides, not polymers. So essentially, polypeptides are essentially many amino acids covalently linked in long chains. In simpler terms, a polymer that consists of two of amino acids as its repeating monomer. Now, basically, the N terminus is basically the amino group of the uh, end of a polypeptide. So it basically acts like a base, and essentially, the C terminus is the carboxyl group of the polypeptide, and essentially acts like an acid. Yeah, that's pretty simple because C stands for carboxyl, and then N just amino. There's an N in amino. That's how you memorize it. And so yeah, that's basically it. And then I also will walk you through those pictures if you want to see them for yourself. Now, protein folding is very interesting. I actually watched an MIT OCW video on protein folding. It was like Fundamentals of Biology, the second video. I recommend you watch it, or second or third. I'm not sure. If you want to put a link, if you want me to put a link down to it, I will. Just ask me. Now, protein folding is basically the process whereby a protein just folds repeatedly in order to get to its functional state, so its biologically active state. Now, the first structure of proteins are primary structure, and this is just the linear array of amino acids making a polypeptide. Now, a lot of people ask, what's the difference between a protein and like a nucleic acid? We haven't gone into nucleic acids, but I'm pretty sure you know what a nucleic acid is, but you don't worry, we'll explain it. But a lot of people ask, what's the difference between like DNA and protein? But the reason why DNA is not a protein is because DNA does not fold to itself to get to its functional state. DNA is hereditary information. Proteins aren't hereditary information. They are required for the transmission of hereditary informations, but they are not hereditary informations. And due to the fact that proteins fold on themselves repeatedly to get to their functional state, differentiates them from nucleic acids, where nucleic acids are just linear chains of nucleotides that never fold to themselves, and they're hereditary information. So that's the difference between proteins and DNA, and why DNA is not a protein and all that cool stuff. So yeah, protein, the primary structure is basically this linear array, like I said. Then the secondary structure is basically more folding from the primary states, and this helps make coils and folds. And there's two types of secondary structures. There's beta plated sheets, which are basically two or more segments from the polypeptide backbone chains. In fact, to be honest, it's a little like um yeah, hold up. I think it was on page 130. It's a little complicated, so let's just look at it because I actually did not like how it was worded over here, and I kind of I usually wrote it okay. So let's just go over here because I didn't really like how it was worded, but this is a picture of them. So basically, alpha helices are basically helices that connect to every fourth amino acid, and then beta pleated sheets are essentially like two segments of a polypeptide backbone which are parallel and formed by hydrogen bonds. So basically, just know they're two secondary structures, and they're quite easily to you can easily tell which one's beta and which one's alpha, and they're both governed by hydrogen bonds. So for secondary structure, just know those two terms, and just know that secondary structure equals more folding from primary structure, which makes coils and folds. And they're essentially just like basically secondary structure is basically focused on like the interactions of the backbone constituents of the polypeptide. And to be honest with you, in our genetics chapter, we'll probably learn more about this. So, like I said, if you're not understanding them, you'll definitely understand them in the genetics chapter. Like chapter one is just a lot of broad stuff, not really that specific. So tertiary structure is basically, I'm just going to keep it simple. It's just basically the interactions between the side chains or the R groups of amino acids. Remember, side chains are essentially just like things that differ the amino acid. Like amino acids differ by their side chains. Now, quaternary structures are basically two of these polypeptides in their tertiary state covalently linked together. Not too complicated. Now, disulfide bridges are basically when like two cytosine monomers. So I think they're two cysteine steine monomers. Essentially, they're sulfur hydro group. A sulfur hydro group is basically a protein stabilizer. So SH. So you should have learned that. Watch my video. I do recommend memorizing the functional groups because they're going to be used throughout this textbook. So get familiar with them. So really memorize them from chapter 4. There's not that much. There's only 7 you have to do. And so basically, like I said, those sulfur hydro groups in the cytosine monomers are essentially brought together by protein folding, and then they're essentially brought close to each other, and then the sulfur of one cytosine bond to another are bridged together. Don't really worry about these ideas, but if you want to see more about cytosine bridges, and look at this. I'm pretty sure. No. It was like protein structure. I wouldn't really worry too much about it. It was actually like a detail. You really need to worry about it. Yeah, don't worry about hydrophobic interaction. I didn't write that down. Those are really not important for this chapter. But yeah, if you want to see the disulfide bridge, then you can. Okay. Now, protein denaturation is basically when proteins lose their shape, which make them biologically inactive. Like if you read the chapter, like literally protein shapes, like are literally what govern the protein's structure and function, like form and function, right? Like but there's so many combinations for proteins and amino acids. So you know, their preciseness of how, like, where the combinations and all that really govern, and their shape and their folding really govern their function. It's really malleable. Like maybe one amino acid switch from to another amino acid could have a major effect in terms of shift of function. It's really intriguing. In fact, this was one of the most interesting. Just proteins are just like they're incredible, like really interesting. That's why I love biology. So nucleotides are basically the monomer of nucleic acids, and they consist of three things: a sugar, which is 5-carbon sugar, a phosphate group, and a nitrogenous base. So when they don't have a phosphate group, they're considered nucleosides. I'm not gonna cover too much about this. Like when we get to our genetics chapter, prefer, preferably for me, genetics is my favorite out of all of them for biology. But when we get to the next chapters, we will be talking about this a lot more. And basically, you need to know purines and pyrimidines. Purines are just adenine, guanine, and they're two nitrogen containing rings. Now, this could be like pretty tricky to memorize. But I use a mnemonic that I actually got from a random video. I don't remember it, so I don't know if I can put the link because I don't remember that video. But the mnemonic I use is pure as gold. So pure, no, not pure as gold. Pure, yeah. Actually, you can put pure as gold, or you can put pure as Ag, because I'll write it out so you can see it. Pure as Ag. No, silver. You know what I mean? So purine, and then for pyrimidine, there's like one nitrogen containing region, and you can just do cut the pi. That's how you memorize it. Pretty sure you can see it for yourself. Now, Chargaff's rule, it didn't actually mention Chargaff's rule, but I was too lazy to say that adenine bonded with thymine and guanine bonded with cytosine, so I just put Chargaff's rule. Chargaff's rule means A is equal to T and G is equal to C, so basically adenine binds with thymine and guanine binds with cytosine. And actually, I shouldn't even put these double lines because that could represent like hydrogen bonds, and there's actually 3 hydrogen bonds in guanine and 2 hydrogen bonds in adenine, but doesn't matter. Phosphodiester bonds are basically bonds that essentially connect nucleotides in one strand together, and basically one phosphate of one monomer of the nucleotide in one strand connects it to sugar groups of another monomer, and that's what phosphodiester bonds are. You can say phosphodiester bonds or whatever. And honestly, like if you just break it down, you can probably memorize it like phospho, then diester, like 2 ester. Like you just can memorize it by yourself. Like if you break it down, it should be. That's why knowing your roots is very important. Now, DNA consists of 4 nucleotides: adenine, thymine, guanine, cytosine, and it's a 5-carbon containing sugar with the sugar deoxyribose. And then for RNA, it's just uracil instead of thymine, and it's a 5-carbon containing sugar. And the important note is that DNA lacks a hydroxyl group on its 2' end, and RNA does not lack a hydroxyl group. Does not lack this. And I do recommend don't memorize these now. Memorize them when you get to the genetics chapter. For now, I wouldn't really memorize all the amino acids and all that stuff, but I think as you move on, you'll definitely pick up to the structures, especially when you start studying biochemistry. So yeah, that's fun. I'm actually gonna do biochemistry like after I do Campbell's. And yeah, that's basically everything for chapter 5. And stay tuned for chapter 6. This was pretty fun. See y'all next time.